Hello, everyone. I'm Donna Snyder from the Office of Pediatric Therapeutics, and I work in the area of pediatric ethics, and I'm here today to talk about ethical considerations for clinical trials. So this is my disclaimer slide. I don't have anything to disclose. And these are our topics for today. We'll talk about the ethical considerations for pediatric trials, because that's my area of expertise, low risk and high risk pathways for pediatric product development, choice of controls including placebo, and this will include considerations for adults, accelerated approval, also including considerations for adults, and a conclusion to my presentation. So this is the basic ethical framework in pediatrics. Children should not be enrolled in a clinical trial unless it's necessary to answer an important scientific or public health question directly relevant to the health and welfare of children. Absent a prospect of direct clinical benefit, the risk to which children are exposed must be low. Children should not be placed at a disadvantage by being enrolled in a clinical trial. And vulnerable populations unable to consent for themselves, including children, should have a suitable proxy to consent for them. Now we'll talk about justification of research risk, and this applies to both adults and children. So for adults, institutional review board approval of research is generally justified by the following criteria. Risk to subjects are reasonable in relation to the anticipated benefits, if any, to subjects and the importance of the knowledge that may be expected to result. However, for children, this criterion is modified in that there's a limit to the risk that knowledge alone can justify, and this is defined in the federal regulations. These regulations are defined under the additional safeguards for children in clinical investigations, or 21 CFR 50 subpart D. We often refer to this simply as subpart D. So research involving children either must be restricted to minimal risk or a minor increase over minimal risk absent a potential for direct benefit to the enrolled child, or must present risks that are justified by the prospect of direct benefit to the child, and the balance of this needs to be at least as favorable as any available alternatives. And we'll talk in a few minutes about what minimal risk and minor increase over minimal risk represent. And of course, we want to obtain the permission of the parent or the guardian and the assent of the child if the child is capable of providing assent. So this slide lists the requirements for children in research as they are defined under the regulations. We've already talked about 50-51, which is minimal risk, 50-52, which is that prospect of direct benefit that's needed in a study if the risk exceeds that minor increase over minimal risk threshold, 50-53, which is the risk is a minor increase over minimal risk with no prospect of direct benefit. And we've talked about the requirements for consent from the parents and assent by the child. What we haven't talked about is this other criteria here, not otherwise approvable, that presents an opportunity to understand, prevent, or alleviate a serious problem affecting the health or welfare of children. The other criteria that I've mentioned can be approved through the IRB process, but this criteria here requires another process, and that process is a federal panel review. So if an IRB determines that a clinical investigation does not meet the requirements under 21, 50, 51, 52, or 53, but believes that the investigation represents a reasonable opportunity to understand, prevent, or alleviate a serious problem affecting the health or welfare of children, the IRB can refer the protocol to FDA to initiate a public review of the protocol by a federal panel of experts. And after consultation with the panel and having this opportunity for public review and comment, the FDA commissioner then can determine whether or not the protocol can move forward, either under the requirements that already exist under 51, 52, or 53, or under this other criteria, 50, 54. And right now, we actually do this process under the Pediatric Advisory Committee process at the FDA. So I also wanted to mention something called component analysis. I talked about these levels of risk and the need for prospect of direct benefit. And when you look at a protocol, any protocol or clinical investigation may include more than one intervention or procedure. And what component analysis asks us to do is to look at each intervention or procedure in a protocol and review them separately to determine whether or not that intervention or procedure does or does not hold out the prospect of direct benefit to the enrolled child. And the reason for doing that is that if we don't carefully look at each one of these interventions or procedures, we may allow a component of a clinical investigation to move forward that exceeds those risk thresholds that I've already defined without a prospect of direct benefit and without referring it to a federal panel. So now we'll talk more about what some of these criteria mean: prospect of direct benefit, minimal risk, and minor increase over minimal risk. So prospect of direct benefit in a protocol means, so what does direct mean? Direct means that it accrues to the individual subject enrolled in the clinical trial, results from the research intervention being studied and not from other clinical interventions included in the protocol. And we often modify the word benefit with the word clinical to indicate that the direct benefit relates to the health of the enrolled subject. So all of this means that when you have a child enrolled in a protocol, you want that child to individually benefit. And we're not looking at the benefits of, say, an additional physical exam being included in the protocol as the benefit of being a participant in that research. We're looking at the benefit, say, for example, from the exposure to the drug in the protocol. And when we think of the prospect of direct benefit, we're not just thinking about whether or not there's proof of concept or there's a mechanistic explanation for why the product might work in children. We also want to look at the structure of the protocol and see if the child might benefit. So we want to have a dose that's appropriate, and we also want to have a study that's long enough where the child has the opportunity to benefit. So I'm just drilling a little bit more into this on this slide. So when we talk about what we need for proof of concept, adult data to support proof of concept may not be needed to initiate studies in pediatric patients. Non-clinical data may be sufficient, especially for conditions that primarily impact children. A minimally effective dose must be tested in a protocol to provide a prospect of direct benefit. Although a lower dose may be safer, if there's no benefit, then testing in pediatric patients is not justified. And finally, the clinical investigation must be of sufficient duration to provide a prospect of direct benefit. This benefit is assessed within the context of the clinical study and is similar to judgments made in clinical practice. And the treatment should be long enough to impact a relevant clinical outcome or an accepted surrogate endpoint, although this accepted surrogate endpoint doesn't necessarily have to be validated for a protocol to move forward under 5052. So what do we mean by low risk? Minimal risk is defined as those risks normally encountered in the daily lives or in the routine medical or psychological examination of healthy children. And then we have this other category called minor increase over minimal risk, which refers to a risk while it goes slightly beyond the narrow boundaries of minimal risk, poses no significant threat to the child's health or well-being. However, it must contribute to generalizable knowledge about the child's disorder or condition. And although this may include children who may be at risk for a disorder or condition. So what does this second criteria mean? So when we apply this minor increase over minimal risk threshold within a protocol, we want this level of risk to only apply to children who have a disorder or condition. So a healthy child could not be exposed to a minor increase over minimal risk. And we want that test to contribute to generalizable knowledge about the child's disorder or condition. So often the question in the protocol is if we, for example, test this particular drug to look at the PK of this product in this condition, might we be able to use that information for a future study to actually study this product in kids where they might benefit. So interventions or procedures within a study that might be considered minimal risk would be a single blood draw, questionnaires, physical exams, or EKGs. And interventions or procedures within a study that might be considered a minor increase over minimal risk might be multiple blood draws, certain x-rays, and we want to look at how much x-ray exposure a child might be exposed to in the study, a single CT scan without contrast, and definitely one with contrast. And then there's finally another type of risk that we might want to think about, which I've already alluded to, that when we have an intervention or procedure within a study that exceeds this minor increase over minimal risk, and therefore we're looking for that prospect of direct benefit to exist, unless the study is allowed to proceed under 5054, that federal panel review option. And the use of an experimental drug or biologic in a study protocol generally falls under those requirements. There are some exceptions. In select cases, a single dose of an experimental drug or biologic with adequate human safety data might be approvable under the 5053 criteria as a minor increase over minimal risk. So an example of that would be where we might do a single dose PK study in a child with a minimal number of blood draws that also meet that minor increase over minimal risk threshold, but we want to have adequate safety information. So oftentimes these types of PK studies are done in children where we already have adult approval of the product and we're just looking for some information to support studies in children. So now we'll talk about choice of control group, and this particular slide applies to both adults and children. So one of the choices is placebo control. This is often considered to be the gold standard. You can also have an active treatment control if you have treatment available. In this case, you want to provide evidence to justify a non-inferiority margin or from previous clinical trials, or you might have a superiority design if you do this active treatment control study. No treatment or non-blinded concurrent control. Dose response concurrent control. And in this situation, you would use different doses within the study. You compare efficacy between those doses to see if you could see a difference. However, if you don't see a difference, it may make it very difficult to interpret the data. And then there's external controls. This includes historical or retrospective controls. And when we do retrospective controls, we usually want to have strong natural history data so that we can make an adequate comparison. And this information comes primarily from ICH E10 choice of control groups, which is an ICH and FDA guidance. So when we're considering the use of a placebo control, there's some things that we need to think about. Participants in the control arm of a clinical investigation should receive an established effective intervention when at all possible. However, placebo or treatment or no treatment may be used in the following situations. And these are defined by the guidances that will appear at the bottom of the screen on the slide. When there is no established effective intervention, or when use of an established effective intervention as a comparator would not lead scientifically reliable results, and when withholding an established effective intervention would not expose participants to additional risk of serious or irreversible harm as a result of not receiving the best proven intervention. So there may be situations where you can use a placebo even if you do have an established effective intervention, but only in this situation. So what about placebo controls in pediatrics? What else do we need to think about? So you may remember we talked about these risk thresholds, and since there's no benefit to children when they're enrolled in the placebo arm of a trial, the risk in the placebo arm should be limited to that minor increase over minimal risk threshold. When we're considering that risk, we need to consider the risk of the placebo itself and the risk of withholding established effective treatment. The route of administration and study duration are important considerations. So giving an oral pill for a period of 2 weeks would have a very different risk threshold than giving an injectable drug over the period of, say, 52 weeks. And we need to determine whether or not we thought that duration or that method of administration would exceed that minor increase over minimal risk threshold. And then we need to think again of the risk from not receiving proven or effective treatment. So if the child is being denied treatment during that time, does that risk of being denied that treatment exceed that minor increase over minimal risk threshold? And these are judgments that are made within the context of the clinical investigation and in the disease process as well. So how can we minimize risk with the placebo control? We want to limit the placebo period to the shortest time needed to establish scientific validity. Placebo controls may be conducted as add-ons trials where all participants receive a specific standard of care, and that can be very important so that they're not denied therapy. Risk may be minimized by adding early escape and rescue treatment for patients with declining clinical status. And we can also consider randomized withdrawal study designs with rescue or time to event. This minimizes time on placebo and sometimes may allow us to collect data on dosing if individuals are randomized to different doses and to placebo at the same time. So now I just wanted to mention briefly accelerated approval because we may be talking about that in the context of this meeting today. So drugs or biologics may be approved that meet an unmet medical need based on a surrogate endpoint or a clinical endpoint that's not based on survival or irreversible morbidity. And this is defined under 21 CFR 314 subpart H. This is the accelerated approval regulations, essentially. When something is approved via accelerated approval, it's approved with restrictions to ensure safe use, and the approval is subject to the requirement that the applicant study the drug further to verify and describe its clinical benefit where there is uncertainty as to the relation of the surrogate endpoint to clinical benefit or of the observed clinical benefit to the ultimate outcome. So this is just a little bit more about accelerated approval. We have this unmet medical need, no therapy exists, or there's a significant advantage of this particular product that's being approved under accelerated approval over available therapy, or this addresses a drug shortage. These may be conditions on which accelerated approval may be granted. They do require phase 4 confirmatory trials, as I've already mentioned. And initially, we do believe that equipoise exists because there's this uncertainty regarding the safety and efficacy of the drug or biologic. We're making some decisions based on this endpoint that we've looked at, but we don't have absolute certainty. And participants in the control arm should receive an established effective intervention if one exists. So in situations where this unmet medical need may fall into this category of a significant advantage over available therapy, then we may compare this product that's been allowed to move forward under accelerated approval. We may compare it to this other therapy that's already been marketed. And placebo may be permissible since treatment is not yet known to result in, to prevent serious harm such as death or irreversible morbidity, as is defined by the regs. But confirmatory phase 4 trials can be challenging, and I'm just including at the bottom of this slide a reference that I found that tends to, that the Friends of Cancer Research at their annual meeting had a workshop where they talked about some of these issues, and they support some of the points that I'm mentioning here. But I think that these are very pertinent when we're thinking about accelerated approval. As a product remains in the market, if benefit is perceived, then equipoise may be harder to maintain. Individuals may be reluctant to enroll in a trial when the product can be obtained clinically or where placebo treatment and an active control may be viewed as inferior. And there may be hesitancy to remove marketing approval if the product may meet an unmet need in a subpopulation, noting that substantial evidence of effectiveness may not exist even in that subpopulation. But there are some potential solutions for conducting confirmatory trials that may help alleviate some of these issues. Starting confirmatory studies in the pre-market setting, and that may allow an interim look at data so we can decide whether or not the product seems to have promise, or maybe even the study can be stopped at that point. Because of the perceived lack of equipoise, a different patient population, for example, one that's less refractory, may be used in confirmatory studies. Real-world evidence may be used to complement results of clinical trials and contribute to the generalizability of the data. And public discussion of clinical trial results to incorporate the patient voice in the risk-benefit assessment may be helpful in terms of moving these studies forward. So finally, in conclusion, children are a vulnerable population, so additional regulatory protections exist for children involved in research. We've covered that in this presentation. It's important to note that research involving children must be either low risk, as we've defined it as minimal or no more than a minor increase over minimal risk, or if the risks are higher, that those risks need to be balanced by the prospect of direct benefit unless reviewed by a federal panel and determined to be ethical and allowed to proceed under that particular regulation. Placebo-controlled trials can be ethically conducted in children and adults if appropriate safeguards are in place. And post-marketing studies conducted after accelerated approval, although challenging, are ethically acceptable. So thanks so much for listening to my presentation, and I hope you all have a great day.